Thank you so much. And we're going to go right into a panel discussion that can include questions for Dr.
Okay.
Kurtzberg about that presentation. If you stay up, Dr. Bradbury, if you can come back down. I'm going to call down again Dr. Ream and somebody I haven't introduced before, Dr. Deepa Rajan, Associate Professor of Pediatrics at the University of Pittsburgh School of Medicine. So we heard about, you could probably all sit on one side, maybe, that'll be easier. We heard about some great preclinical work being done, and we also heard about the difficulty and frustration of dropped clinical trials from the standpoint of financial, business kind of reasons on these companies. When a group of us got together several months ago to think about the state of Krabbe for the Global Leukodystrophy Initiative meeting in February of 2025 in Philadelphia, we talked about what could be potential steps. I think solutions maybe is too strong a word, but at least steps towards improving this situation. One thing that was brought up was the idea of creating some kind of unified clinical trial design or outcome measure for these trials. So if there were multiple companies that were either dropping or starting, et cetera, that there would be some unity to what is being done for these patients. So what I thought we could just kind of briefly brainstorm with this excellent group and then also our audience here is how can we move forward with this problem of great preclinical work being done and difficulty translating to clinical trials? Is there some opportunity for group to get together and think about unified trial design and any other sort of steps that we think that we can take to accomplish what Dr. Kurtzberg just said, which is basically getting these therapies to clinical trials. So that's the question for this brief panel discussion, but also if anybody has any initial questions for Dr. Kurtzberg about the data that she presented, why don't we start with that?
Thank you. Great presentations by everybody. I'm Stephanie from Atlanta again. I just had a question on the last case that you showed, Dr. Kurtzberg, about the patient who received the transplant and the gene therapy. Since that child's still obviously now has some disabilities, how does that compare for you to the children that just get transplant?
He's better than most of the children I've seen. Tara's seen him too. He's doing more and he's doing it more quickly. And his tests have been more stable than I would have expected.
I have a question. For those, back to the very beginning of your talk with the 2 transplant alone cases that were very different, was their conditioning the same and was their engraftment similar?
Yes. They had exactly the same conditioning and GVH prophylaxis, and they both engrafted early, which is usual for babies getting transplanted because they get a high cell dose. Yeah. Yeah, I don't know how to predict yet.
I also have a question for you for the CP study you were talking about. There was something on the slide about biomarkers and you listed epigenetic markers. I wonder if you could go into that a little bit more.
I can sort of, there's a guy at Nemours Children's who is looking at epigenetic markers on newborn blood spots to detect kids with CP, and they have a big neuromuscular disease program there. And I mean, I have to tell the story. So the wife of E.I. DuPont in the late 40s, she was his 4th wife, wanted a present. And he said, "What do you want?" And she said, "I want to build a children's hospital on our estate for kids with polio." And they built this hospital that ultimately became Nemours Children's in Wilmington, Delaware. And they have an amazing motor development program. Anyway, so we were talking to them, and I'm blanking on the guy's name, it'll come to me. But he went and took all the kids who presented in their CP clinic at the age of 5 or 6 and went back and looked at epigenetic findings at birth and found he could detect kids with no clinical history that would have predicted CP. So we were We wrote a big ARPA-H grant that didn't get funded, but in that grant, we were going to validate his observations and move it forward. So it's part of that study.
All right, great. So moving on to our panel question, the big question is, what do we do? That's the big question. How can we get this moving forward in terms of translation of preclinical to clinical? And maybe the more specific question is, how would we go about modeling what some other leukodystrophies have done in terms of coming out with unified clinical trial design outcome measures?
I'll start. I mean, I think we should do what they did with COVID. We should have a warp speed approach. We should do, no, I mean, we should we should agree on endpoints, and the endpoint should be short term. And I think to your question, with the gene therapy, it really looks like nerve conduction and peripheral nervous motor function is better. So use that as an endpoint at 3, 6, 9 months, something like that, and screen a few different approaches. So it might be transplant alone or transplant plus some kind of gene therapy, systemic or gene therapy intrathecal, or both together. And sorry, go that way. And you have a clear endpoint that's go, no go. And you bring the promising, so it's like a pyramid. You bring the promising ones down and try them again. Now, how to commercialize it, how to fund it, that's really the problem. We either need somebody, like Bezos to get us interested in this disease, or we need to get ARPA-H to fund a big program with the idea that 50 years from now, we'd have an approach that could fit for adults who get whole genome sequencing when they're born and they're going to develop neurodegenerative disease X, Y, or Z. We're going to figure it out in these kids so we have a therapy for them 50 years later. I think we might be able to talk ARPA-H into that. Anyway, that's all I can think of.
I agree with the need for streamlined outcome measures, because with the Forge, what was going to be the neurogene and the Passage trial, the outcome measures were not the same. So although all of the interventions were different between which serotype, the route of administration, and the protocol, so it was already looking at 3 different approaches, and then with 3 different primary outcome measures, it was going to be very difficult at the end to say which trial was the most efficacious. And so I like what the Vanishing White Matter community did when they came up with a consensus paper for outcome measures. So if you were looking at different small molecules or eventually a gene therapy, everyone had a consensus on which outcome measure was going to be used for the trial. And what they further did was they stratified it based on if it was infantile, late infantile, juvenile, because sometimes the outcome measures are going to need to move based on the presentation of the disease. So I would love to see Krabbe do something like that, like if we're going to treat early infantile or juvenile patients, we would use nerve conduction would be primary. And if we could all come to a consensus and then basically force the companies to say, okay, this is what the thought leaders already want to see. And then there's not a huge decision to be made. And then there could be a little bit of wiggle room on secondary outcome measures, but at least if a few of them were the same, we would be able to know at the end of the day, which, if we're comparing nerve conduction versus nerve conduction, we know that, we were moving the needle.
Remind us, what were the outcome measures that the 2 different trials were using, or does anybody remember?
Well, Passage was primarily safety because it was a phase. It was safety. It was a phase 1, and it was escalating by moving shorter. So it was 9 to 12-month-olds, 6 to 9-month-olds, 3 to 6-month-olds, and newborns as their phase back, giving the same dose, interforamen magnum to each kid. So of course, they only did that last oldest cohort, 1 kid. That was their design. And they had a lot of secondary endpoints with MRI and nerve conduction, all the usual stuff, but that was not their primary. Neurogene really didn't get that far. Yeah. And Forge, I'm confused about what the outcomes are with Forge. So, because it's a big bucket of stuff. Yeah.
Just looking at the data from Forge, I don't know what so these kids were transplanted, and then they got the AAV, right? We did all the cyclosine and Galci. The cyclosine on all of the patients, were all below 4 nanomolar. So, I suppose some of these kids, when they were born, had much higher, but we see that transplant alone will lower cyclosine. I didn't see cyclosine go down significantly after AAV, but it was low. It was the highest babies were 4 nanomolar. I saw Galci is high in the blood, but they got transplanted, so of course it's high. In the animals, when you add AAV, the Galci in the liver explodes. It's 100 times higher. Everybody knows it goes to liver. There's no Galci in CSF. We measured it. It's 0. Urine 0. So the only place we measure Galci is in the blood, and they got transplanted. Of course, they have high Galci in the blood. So I didn't learn anything by measuring Galci, and I didn't learn anything by measuring cyclosine. It didn't go down. It was low to begin with, and I can only imagine it was high in some of these patients before they were transplanted, but I don't have that data. So it's been really hard from Galci and cyclosine to untangle transplant from AAV. So I think it's going to come, it's exactly what Allison and Joanne are saying. It's going to come down to these neurological tests and clinical assessment. It's not going to come down to cyclosine or Galci, I think, if you do it on top of transplant, right? Allison, I mean, I can tell you the cyclosine is not going down, but it's never very high in these patients. In reclaim and rescue, it's always low. So I don't know what to say.
But I don't disagree with you, and that's more data than I know
Yeah.
from those studies, except our one patient. But you're getting their post-transplant initial sample, so.
Yes, exactly. So, but the transplant, I bet, is doing most of the cyclosine drop. I don't see it go down after AAV. I have the baseline before
Yeah,
AAV.
right.
It's low, and it doesn't go down with AAV significantly.
Okay.
It doesn't go down further with AAV.
Yeah, and
But it was low, so I don't know what to say. If you did AAV alone, I bet the cyclosine would go down, but I can't see that on top of a transplant.
our one high-dose patient, Paul sent clinical testing on cyclosine to Dieter, and the last one was 8 in red cells, and the enzyme level is sort of in the mid-range of normal enzyme. It's not, this is blood enzyme, but not higher than, I don't know, I was led to believe it would be ultra high, and it's not.
I was going to add, just with the transplant studies, the long-term outcome, the peripheral neuropathy seems to catch up with the children more than 5 years after the transplant, right? And so like having a clinical trial, doing something that is transplant with gene therapy and then following up on peripheral neuropathy and expecting to see outcomes within 5 years, I think we are setting ourselves up
For failure. I mean, certainly the more recent transplants I'm aware of, the ones we reported from the 4 institutions, none of those kids walk. They never walked. They're all transplant alone, but all done roughly first 5 weeks of life. And none of them walked. And they're varied in terms of their other abilities, but we now have like 5-year to 7-year follow-up. But they never walked independently. So I really think you can use that as a
I would
very
agree.
simple.
I think
Yeah.
most of our children post-transplant, but again, that's 15-year follow-up data, they don't walk.
No, I mean like 1 year, 2 year. I know. I mean, I'd love to do 3 months, but I'm just saying I don't know if my little guy, the gene therapy guy, is going to walk, hopefully, but he's 15 months now. But the other kids that we've studied in the last 7, 8 years, who all got neonatal transplants, never walked. So I think you said, do they walk by 18 months or 15 months or whatever you want to? There's ways we could talk about what's the best time frame. But I think we have that.
Yeah, I mean, I would go back to what you mentioned about a registry, right? Like there are certain things we cannot control, but what is it that we can do? I think we have rich databases of data collected for these children for 20 years, but they're in specific silos,
Yeah.
right? And maybe what we need to do is to work on unifying that. And there's a lot of unanswered questions, like why is it that we cannot predict who does well with transplant? Is there a risk stratification in that retrospective data that we could do? And then we all talked about there is a diagnostic biomarker for early infantile with psychosine, but no kind of predictive biomarker for the late onset disease. So I feel like the things that we could do is to work on the data that's there and help stratify disease and stratify risk better.
Yeah, I agree with you. I didn't mention, but I put a grant into the NMDP to try to get them to fund a retrospective/prospective transplant outcome study. And they didn't fund it, but they did work out a way for me to get all the retrospective data from the transplant registry, which is called the CIBMTR. So my IRB just signed off on the data transfer, but I will be getting that download. So anything that's in the CIBMTR database, which doesn't have great descriptive neural outcomes, but it has transplant outcomes for as long as the patient lives, we will have. So that's a start. But it's coming to me like in a red, I can put it in whatever I want. So probably a red cap because we built one for Neurogene that they didn't use. But anyway, that's a start. And I know, Paul, don't you have some funding for a registry? So we all have to talk because we don't need more than one registry.
So I was gonna just say, I think that's the walking is the clear obvious, because why would you be doing this instead of transplant? And the main reason it sounded like was the walking. And so I think that the walking makes by far the most sense. And I think the FDA wants to see something that's clinically meaningful more than just a change. I know right before COVID, we had met with ALD Connect with some wearable device people, and it was unfortunate timing because then everything shut down like the next day. But I do think, and I know Dr. Nagy, who had to step out, has done some with our Canavan kids, but I think these wearables are getting better. And if you could measure how much someone's walking and then also balance, that would maybe give you some more objective type of data. We've even looked at doing things like that with the eye tracking to see for some of these kids that are more severely. But I do think that the wearables, we don't take as much advantage of as we could.
I just wanted to have, this is Laura from neurology at CHOP. I just wanted to have 2 points. I think as we're starting to move into trial design for these ultra rare disorders, I worry a little bit about us shooting ourselves in the foot. We're jumping really fast into efficacy outcomes, whereas if we could demonstrate a positive safety trial and tolerability trial, then we would have something positive in a small number of children and then quickly transition that into a second trial. And by really us as a rare disease community consistently trying to make things faster and easier, I think we're failing to show benefit when we really could. And then we could be very thoughtful and put a panoply of outcome measures as exploratory outcomes, the way clinical trials were originally intended for us to do, is to do it in a stepwise way. We're trying to jump to step 3. And you're still going to treat the same number of kids though, right? You're just going. I hear you and I hear your point. I will respectfully have a different perspective, and I've changed. I used to be, we need to go straight into the efficacy, but I think we're really failing to demonstrate safety and tolerability as a phase 1 in a small number of kids. We're also using biomarkers in a way they haven't been validated. They aren't surrogate biomarkers by formal criteria. We haven't shown that. And so by us leaning too heavily on them, I think we also are either getting false confidence or we're falsely shooting ourselves in the foot. I will pull out the lovely design Takeda trial where they did intrathecal enzyme infusion into kids every single week. They saw dramatic improvement of sulfatides fairly immediately, and they saw absolutely no benefit clinically, right? They fixed it, but they fixed it in the wrong compartment. And so I think as we're measuring biomarkers, they need to really be validated for what we're using them for too.
So Paul Orchard from Minnesota. So I just want to take a step back a little bit. So how are we going to move this field forward? I mean, in terms of novel therapy, it seems to me that catch-22 kind of is that these are rare diseases. It's tough for industry to make a buck doing this. And so, you know, because some of these clinical trials take 10 years and cost $500 million to do before you get them licensed. Unless there's a payoff, they don't want to do it. And I get it. I think the academic world, you know, CHOP or Minnesota or Boston or Kennedy Creek or whoever might want to develop novel therapies. But if we use the same yardstick where we have to come up with $300-400 million, that's not going to happen either. Is there some way we can go to the FDA and have a different paradigm set up so we can develop some therapies for these diseases that's not using the same benchmark that we've been using historically? Because otherwise I'm not very optimistic that a lot of these really rare diseases we're going to find any sort of therapy for. So, I don't think anybody was here, we got like 3 minutes. Anybody here is going to fix this in the next few minutes, but I don't know, maybe Anna or the advocacy groups need to help bring these things back to the FDA and say, look, this isn't working. I mean, the companies are pulling out and the academic institutions can't do it on their own. We need a different pathway to make these things work so it can offer something for these families.
I mean, I will say with the CAR T therapy, which is slightly different, which was developed at CHOP, they have a perpetual IND open, right? So they're not looking for approval, they're looking for access. And I think if we set our goals differently, at least until the system changes, that's one option too.
Yeah, but CAR T, it's a much bigger market and much easier to make and much easier to test because your endpoint initially was, you know, kids with relapse leukemia were going to die, and they didn't. They went in remission, right? So that's like the prototype for how you get oncology drugs through. And CHOP has gotten a ton of money for CAR T, which is great. Should happen to all of us. But it's not the same as having 4 kids with Krabbe in the whole country in a year, which is kind of what we're looking at.
One thing going back to the safety, I don't know if you were also, are you wanting like a dose escalation?
Yes,
Yeah.
please. I mean, I feel like we can't give up everything, right? We're saying we won't placebo control, which I agree with. Like, we have to have some level where we've done it in a staged way. So I think a safety with dose escalation is a very reasonable first step.
Yeah, and both the Passage and the Forge trial were dose escalation trials. For our CSF trial, we met with the FDA specifically about this in our pre-IND, and we said the data is very clear in the mice and the dogs that if you underdose them, you just put them in this attenuated disease state that we didn't feel was ethical, because then you're knowingly underdosing them and they're going to live longer, but they're just going to have this, horrendous version, of the disease. And the FDA was actually in agreement and said, you can start at your high dose, and as long as you're able to show safety at your high dose, then you don't have to go down. So I thought that was reasonable that the FDA was willing to work with us on that. Of course, we're going to look at all the traditional safety parameters, but there's also that balance of doing something that is not in the best interest of the kid just to get a readout.
Any other questions or comments?
And I think part of the problem with Krabbe is that you're in this situation where you have a treatment, right? If there's no treatment, like with the Tay-Sachs trial, right? It was easier to, you know, it didn't work, but it was easier to do because there was no option. Whereas in this case, you're trying to decide, you know, do you go with the traditional, do you go with, and I know we had someone come in and talk to our group at MGH about this umbrella method, and something about like the, you know, rain is getting all caught by the umbrella. And my original thought was like, there's no way parents would agree to this, and it was you'd randomly have been assigned. But for the ALD gene therapy, one of the points was it would have helped us a lot, because had we done the original 2 Blueberg trials that way, we would have had some kids getting one chemo regimen, some kids getting another chemo regimen, and they would have all been getting the same whatever you call it, vector, right? And so we would have had fewer things. So maybe it's changing the narrative so families are open to this idea that you're going to randomly be assigned to either traditional transplant, transplant with gene therapy, or just gene therapy, which I understand nobody likes. But I know when they were discussing it with us, there is an argument for how that would have, in the long run, I think potentially have helped us a lot. No, you know, is it one of the promoters, or is it gene therapy just is going to cause these? And now we can't get that back. So maybe it's talking to the family groups in part.
I mean, I agree. The family groups need to have a seat at the head of the table at all times. That's, you know, very clear. But in a way, I mean, even if we don't do this adaptive trial design, some families are still going to choose transplant. The transplant plus gene therapy door is closed unless someone else picks up the Forge trial, which there's no interest right now because it was just a very complicated trial design. And then we're hoping to get the gene therapy alone to the clinic. We just don't know when that's going to happen, but there's decades of transplant alone data to compare to. There's untransplanted kids to compare to. So that natural history data already exists, and there will definitely be families that still choose transplant and definitely families that choose nothing. And then you're going to have sib sets too, where it was just too late that the sibling gets one or the other.
And we're out of time, except for one last comment.
sorry.
Dr. Wu.
No, I wasn't watching the time. So I think involving families and advocates early in the, you know, the outcome measures is super important. And I think the, you know, clinically meaningful outcomes are they walking at 18 months or 24 months? That's great, but that might not be the most important thing, especially if we're maybe not expecting them to really recover their neuropathy fully. So other strength measures, other function measures. So Allison had brought up involving advocates, and to bring us back full circle to the beginning question, we need to involve them at all stages.
All right, great. Thank you so much. That was a great discussion, and I'd like to propose that Hunter's Hope maybe put together a work group among interested parties to discuss this idea of outcome measures, unified outcome measures, to see if we can sort of move those ideas forward.